first of all, the need for reduction division during meiosis. Okay, right. So basically, we know what happens is that in fertilization, the nuclei of gametes is going to fuse together, right? Now, gametes are going to have haploid number of chromosomes, right? That means they're going to have half the number of chromosomes, right? So basically, what's going to happen is that when in fertilization, our male gamete and female gamete are going to fuse, so the original diploid number, which is specific to the species, will be restored, right? So meiosis is basically a nuclear division that is going to reduce the chromosome number of a cell, and we know that in humans the chromosome number is reduced from 46, which is diploid, to 23. That is haploid, right? So basically that's the need for reduction division during meiosis. Now moving on to homologous chromosomes. Right, so basically what happens is that in diploid cells there are two complete sets of chromosomes in the nucleus, right? Now homologous chromosomes are going to have a characteristic shape, they're going to have a fixed length, and the position of the centromere will be in a particular location, right? Now what happens is that basically in homologous chromosomes, another thing to note is that they're going to carry the same genes in the same positions, right? For example, homologous chromosomes, right? So that is, we're talking about a pair of chromosomes, right? And each pair consists of two sister chromatids joined at a centromere, right? So that is what we're talking about here, right? Now what is going to happen is that during fertilization, we know that a diploid zygote is formed, right? So in a zygote, one chromosome, I just explained you that we have a pair of chromosomes. That means one chromosome, that is two sister chromatids joined at a centromere coming from the mother, and the other one coming from the father, right? That is, one is the female gamete, one is coming from the female gamete, and the other is coming from the male gamete, right? So having the same genes in the same order is going to help the homologous chromosomes line up alongside each other during meiosis, right? Now moving on to the next pointer, meiosis in animal and plant cells. Meiosis is a form of nuclear division that results in the production of haploid cells from diploid cells, which we just studied, right? And we know that it produces gametes in plants and animals that are used in sexual reproduction, and it's going to have many similarities to mitosis. However, it has two divisions, right? So when we'll be talking about meiosis, we'll be talking about two divisions, right? Meiosis 1 and meiosis 2. And each of these divisions, we're going to have the stages. For example, when I'm talking about meiosis 1, then prophase 1, metaphase 1, anaphase 1, and telophase 1, right? And then when I'm talking about meiosis 2, then all of these stages are going to get a 2 in front of them, right? Now when I'm talking about prophase 1, what's going to happen is that our DNA is going to condense and it's going to become visible as chromosomes, right? And DNA replication has already occurred, so your chromosome will consist of two sister chromatids joined together at a centromere, right? Now what's going to happen is that the chromosomes will be arranged side by side in homologous pairs, right? And this pair of homologous chromosomes is what we call a bivalent, right? Now because these sister chromatids are very close together, right? Just imagine two sister chromatids joined at a centromere, and then two of those, meaning a pair of homologous chromosomes. Now they're very close together, right? So what's going to happen is that there might be an exchange of parts between those two non-sister chromatids, right? And so what's that going to do is it's going to produce a new combination, right? So, and another thing that's going to happen in prophase 1 is that the nuclear envelope is going to break down, the nucleolus is going to disintegrate, and the centrioles are going to migrate to opposite poles, and the spindle is formed, right? Okay. Now, another point to note is the point that I was telling you about the exchange of parts. It's called crossing over, and the point at which it occurs is called chiasma, right? And the plural is going to be chiasmata, right? Now moving on to metaphase 1. Okay, so our bivalents, which are basically pairs of homologous chromosomes, they're going to line up along the equator of the spindle, right? and what's going to happen is that the spindle fibers are going to get attached to the centromeres. Now remember, here we're talking about a pair of homologous chromosomes, and that as I have been explaining, that we have two sister chromatids joined at a centromere, that is one chromosome, right, and then a pair of them, right. So when our spindle fibers are going to extend, they're going to join to the centromere of each chromosome, right, which exists of two sister chromatids, right. So when they're going to pull away, they're ideally pulling two sister chromatids joined at the centromere, right. So that is why we're going to get haploid number, right. So okay, and this part of pulling whole chromosomes to opposite ends of the spindle occurs in anaphase 1, right? And the centromeres are not going to divide, right? In telophase 1, what's going to happen is that, of course, our chromosomes are now at opposite poles, so our spindle fibers are going to start breaking down, and nuclear envelopes are going to form around the two groups of chromosomes, right? And now, some plant cells go straight into meiosis 2 without reformation of the nucleus in telophase 1, right? But here we're going to have the reformation of the nucleus because we're talking about animal cells, right? Now, cytokinesis is where the division of the cytoplasm occurs, right? So what's going to happen is that the cell organelles are going to get distributed between the two developing cells, and a cleavage furrow is going to form in the middle of the cell, which is going to contract to divide the cytoplasm in half, while the opposite occurs in plant cells, okay? Basically, what's going to happen in plant cells is that the vesicles from Golgi apparatus, they're going to gather along the equator of the spindle, which is basically the part at which our homologous chromosomes arrange themselves, and the vesicles are going to merge with each other to form the new cell membrane, right? So, and that is going to, and they're also going to secrete a layer of calcium pectate, which is going to become the middle lamella, right? And layers of cellulose will be laid upon the middle lamella to form the primary and secondary walls of the cell, right? So this is what's happening in cytokinesis. This is how it's different in plants and animal cells, and you need to remember this, right? Now, the end product, of course, is going to be two haploid cells, as we have been talking about, right? Right? Now, what happens in second division is that basically there's not going to be an interphase, meaning there is no DNA replication, right, taking place. Now, in prophase 2, again, we're going to have the same idea happening again, that the nuclear envelope is going to break down, the chromosomes are going to condense, and the spindle forms at a right angle to the old one, right? Okay, in metaphase 2, again, we're going to line up along the equator of the spindle. Now, what you need to remember here is that instead of our homologous pair of chromosomes, right, now here we just have a chromosome, meaning just two sister chromatids with the centromere, right? So when our spindles are going to attach to the centromere, they'll be pulling the two sister chromatids apart, right? So the centromere, no more centromere is going to exist between them. That is where we'll be getting 4 cells here, right? So that you need to be clear with, right? Okay, now, and this thing of pulling apart always happens in anaphase, right? And in telophase 2, simply your nuclear membranes form around, right? See in here, in telophase 1, again, nuclear envelopes were forming around, the spindle fibers were getting broken down, and in telophase 2, again, our nuclear membranes are forming around each group of chromosomes. Then again, cytokinesis, and this time you're going to have 4 cells, right? Okay. Now what's happening here is, okay, the cells still contain the same number of centromeres as they did at the start of meiosis 1, but they now only have half the number of chromosomes, right? Previously chromatids, right? So I hope you understand this point now, right? So now there are 4 haploid cells, right? Okay, now moving on to meiosis 1 and meiosis 2, how you can differentiate between them, right? Homologous chromosomes pair up side by side in meiosis 1 only. This means if there are pairs of chromosomes in a diagram or photomicrograph, meiosis 1 must be occurring. The number of cells forming can help distinguish between meiosis 1 and meiosis 2, right? And if there are 2 cells forming, it is meiosis 1, and if there are 4 new cells forming, then it's going to be meiosis 2, right? Now, the distinguishing features are that in prophase 1, again, I told you, homologous pairs of chromosomes, right? Metaphase 1, homologous pairs of chromosomes are going to line up, right? Anaphase 1, whole chromosomes are going to be pulled up to opposite poles, right? Telophase 1, there are 2 groups of condensed chromosomes around which nuclear membranes are forming, right? Cytokinesis, cytoplasm is dividing and cell membrane is pinching inwards to form two cells, right? In prophase 2, we can see single whole chromosomes, right? Not homologous pairs, right? In metaphase 2, we have the whole chromosomes lined up along the equator of the spindle, not a pair of homologous chromosomes, just single whole chromosomes, right? In anaphase 2, the centromeres are going to divide and chromatids are being pulled to opposite poles, right? Telophase 2, nuclei are forming around the four groups of condensed chromosomes, right? And cytokinesis, cytoplasm is dividing and four haploid cells are forming, right? So here you can also see in the picture, this is meiosis. This is meiosis 1, right? In the starting, right? So you can see that whole pairs are lining up along the equator, and our whole chromosomes are being pulled, right? While in our anaphase 2, you can see just single chromatids are being pulled, right? So that is why we're going to have four haploid cells, right? So you should be able to look at the diagram and identify them in paper, right? So you'll identify by the distinguishing features we just studied, right? Okay. Now, meiosis, sources of genetic variation. Having genetically different offspring can be advantageous for natural selection. Meiosis has several mechanisms that increase the genetic diversity of gametes produced, right? And okay, both crossing over and independent assortment, random orientation result in different combinations of alleles in gametes, right? So this is also a question you can get, right? Now, what you need to remember is, first of all, that how can genetic variation occur, right? Genetic variation can occur, first of all, the mating is completely random, the fertilization of gametes is completely random, any female gamete can fertilize with any male gamete, right? So that's completely random, so that's already producing genetic variation, right? Then considering these processes, what's producing genetic variation are two other things. We have crossing over, and we also have independent assortment, right? So crossing over, I just told you, it's the process in which non-sister chromatids are going to exchange alleles, right? And that was occurring in prophase 1, right? So during meiosis 1, homologous chromosomes pair up and are in very close proximity to each other, right? So as you can see in our prophase 1, right? So prophase 1 is basically that thing you can see, right? In the diagram, right? So in that you can see that the chromosomes, the homologous chromosomes, because they have very close proximity, they may exchange parts, right? And these crossing points will be called chiasmata, right? The entanglement places stress on the DNA molecules. As a result of this, a section of chromatid from one chromosome may break and rejoin with the chromatid from the other chromosome, right? And this swapping of alleles is significant as it can result in a new combination of alleles on the two chromosomes, right? There is usually at least one, if not more, chiasmata present in each bivalent, which is your pair of homologous chromosomes, right? Crossing over is more likely to occur further down the chromosome away from the centromere, right? So this is just a point that you need to remember, right? Now, moving on to independent assortment. What is this, right? Okay, so independent assortment is the production of different combinations of alleles in daughter cells due to the random alignment of homologous pairs along the equator of the spindle during metaphase 1, right? So during metaphase 1, we just did it, right? And we knew that homologous pairs were lining up, right? Now, any homologous pairs might align with each other in any random manner, right? And so when our cytoplasm is gonna divide, right? So each cell can have a different combination of chromosomes, right? And this is what independent assortment is, that the alignment is not dependent on anything. It's very random, right? So the different combinations of chromosomes in daughter cells increases genetic variation between gametes. In prophase 1, homologous chromosomes pair up, and in metaphase 1, they're pulled towards the equator of the spindle, right? So each pair can be arranged with either chromosome on top. This is completely random, right? The orientation of one homologous pair is independent, unaffected by the orientation of any other pair, right? So the homologous chromosomes are then separated and pulled apart to different poles. The combination of alleles that end up in each daughter cell depends on how the pairs of homologous chromosomes were lined up, right? So, to work out the number of different possible chromosome combinations, we can use 2n, right? So that means 2 to the power 23, right? So that gives you 8,324,608 different combinations, right? That is how, to the extent our independent assortment can produce genetic variation, right? We are moving on to the next pointer, fusion of gametes. Meiosis creates genetic variation between the gametes produced by an individual through crossing over and independent assortment. This means each gamete carries substantially different alleles, right? Okay, so basically during fertilization, I just told you that any male gamete can fuse with any female gamete to form a zygote, right? And this kind of fusion of gametes at fertilization creates genetic variation between zygotes, as each will have a unique combination of alleles, right? There is almost zero chance of individual organisms resulting from successive sexual reproduction being genetically identical, because we just looked at the different combinations that can be made, right? Now, genes and alleles, right? So, the DNA contained within a chromosome is essential for cell survival. Each chromosome consists of a long DNA molecule which codes for several different proteins, right? We have been doing this, right? And a section of DNA which is going to code for a single polypeptide or protein will be called a gene, right? And the position of a gene on a chromosome is its locus, and plural is loci. And we just did it that alleles are going to be arranged on the same loci, right? But they can code for different things, right? For example, alleles are basically different forms of gene, right? But the same gene. For example, hair color, so the alleles can be brown, black, blue, right? Like that, right? Now, each gene can exist in two or more different forms called alleles. Different alleles of a gene have slightly different nucleotide sequences, but they still occupy the same position on the chromosome, right? That is what I was doing. Now, moving on to genotype and phenotype. The chromosome of eukaryotic cells occur in homologous pairs. There are two chromosomes of each chromosome. There are two copies of each chromosome, right? As a result, cells have two copies of every gene. As there are two copies of a gene present in an individual, that means there can be different allele combinations within an individual, right? So basically, when we're talking about genotype, we're talking about the alleles of a gene possessed by that individual, right? Basically, we're talking about the different forms of the gene, right? The different alleles can be represented by letters. When the two allele copies are identical in an individual, they're said to be homozygous, right? And when those two copies are going to be different, they're said to be heterozygous, right? And we're going to represent them with different letters, right? So ideally what we do is that we represent the dominant allele by our capital H, right? Not capital H, the capital letter, right? And our recessive allele with the small letter, right? And because they're alleles, different forms of the same gene will use the same letter, right? Now, phenotype will be the observable characteristics of an organism, right? So basically, phenotype is anything that we're able to observe, right? And genotype refers to the particular combination of genes we're talking about for an individual, right? Now, moving on to F1, F2, and test crosses. When a homozygous dominant individual is crossed with a homozygous recessive individual, the offspring are called the F1 generation, right? So, homozygous dominant individual, that means that we're going to have two capital letters, right? So, for example, AA, right? It's crossed with a homozygous recessive. So, the same letter but smaller, right? So, AA but small a, right? The offspring is going to call F1 generation, right? And all of the F1 generation are going to be heterozygous, meaning if I crossed two capital A's with two small a's, I'm going to get what? I'm going to get one capital A, one small a. That is heterozygous, right? So if two individuals from the F1 generation are then crossed, the offspring that they will produce will be called the F2 generation, right? Now, a test cross can be used to try and reduce the genotype of an unknown individual that is expressing a dominant phenotype, right? So basically, we can use a test cross to identify the genotype of an unknown individual, right? And now what we can do is that basically what the individual in question will be crossed with an individual that is expressing the recessive phenotype, right? So what's going to happen is basically what we're doing here is that we're using test cross to identify an unknown genotype, right? And how we're doing is that basically we are crossing the individual with the unknown genotype with an individual that is expressing the recessive phenotype, right? Now what's going to happen is that the resulting phenotypes will provide us sufficient information to suggest the genotype of the unknown individual, right? Now if the offspring, right, is expressing any recessive phenotype, then the unknown individual is heterozygous. That's what you need to know, right? So what we talked about in under this heading is that basically when we're talking about crossing homozygous recessive individual with the homozygous dominant individual, the offspring are called F1 generation, and when we're going to have two individuals from the F1 generation cross, they're going to produce the F2 generation. Now if we have basically an unknown individual, unknown genotype of an individual, then we can cross it with a recessive with an individual of recessive genotype, and if as a result we get any recessive genotype being expressed, then we should know that the unknown individual has a heterozygous genotype, right? Now moving on to linkage. There are two types of linkage in genetics: sex linkage and autosomal linkage, right? So basically we know there are two sex chromosomes, X and Y, right? Women are going to have XX, whereas men are going to have one X chromosome and one shorter Y chromosome, right? So basically what happens is that some genes are found in a region of a sex chromosome that is not present on the other sex chromosome, right? So basically sex-linked diseases, right? For example, right? So as the inheritance of these genes is dependent on the sex of the individual, they're called sex-linked genes, right? Most often, sex-linked genes are found on the longer X chromosome. Hemophilia is a well-known example of a sex-linked disease, right? So basically, what happens is that sex-linked genes are represented in the genotype by writing the alleles as superscript next to the sex chromosome. For example, a particular gene that is found only on the X chromosome has two alleles, G and G. The genotype of a heterozygous female would be written as XG, right? So that's in the superscript, right? And a male's genotype will be written as XGY, right? Because it's not present on the Y chromosome, right? So if the gene is on the X chromosome, males will only have one copy of the gene, whereas females will have two, right? So basically, what's happening here is that basically if we're talking about hemophilia, right? So hemophilia is sex-linked disease. It requires the X chromosome only, right? So if it's on the X chromosome of a male, then the male definitely has hemophilia, right? If the person does not has the gene on the X chromosome, then he does not have hemophilia. But the case is different with the female, because female has both X chromosomes, right? So what's going to happen is that the female might be a carrier, right? Because she might be heterozygous, or she might be homozygous, right? So only in the case of homozygous will she have hemophilia, right? So she might be normal, but she might have, when she is heterozygous, she will definitely be a carrier, right? So she may express one of the three phenotypes. She might be normal, carrier, or might have the disease, right? Now we can see a worked example also. So hemophilia, we just talked about it, right? There is a gene found on the X chromosome that codes for a protein called factor VIII. Factor VIII is needed to make blood clot. There are two factors of factor VIII, the dominant F allele, which codes for normal factor VIII, and the recessive F allele, which results in a lack of factor VIII. When a person possesses only the recessive allele, they don't produce factor VIII, and their blood can't clot normally, right? So it just requires the recessive allele to result in a lack of factor VIII, right? The genetic diagram below shows how two parents with normal factor VIII can have offspring with hemophilia, right? So again, we can see here that the parents have what? They have normal factor VIII, right? So we can see that they both are. The female is heterozygous and the male has a capital F, meaning that we have a normal factor VIII, right? So when we're going to cross both of them, we can see here what's happening is that we have female with normal blood clotting, we have male with normal blood clotting, but we also have a male with hemophilia, because we just read about it that we only required recessive allele to have hemophilia, right? So that is why when it's going to be on a male and a male receives even one recessive allele, he is going to confirm have hemophilia, right? Now this was one type of linkage, right? Now moving on to the other type of linkage, which is autosomal linkage, right? Now, what is autosomal linkage? Autosome is any other chromosome other than the sex chromosome, right? So basically, two or more genes on the same chromosome do not assort independently, right? Because why? Because these genes are linked and they are going to stay together in the original parental combination, right? So linkage in between gene affects how parental alleles are passed on to offspring through the gametes, right? So basically, autosomal linkage, you need to remember 3 pointers for it, that basically autosomes are chromosomes other than the sex chromosomes, right? And the genes are going to be linked, so they're going to stay together in the original parental combination and they're not going to sort independently, right? That's all you need to remember, right? Now, we also have a birth example for this, right? Now, what you can see here is the genes for tail length and scale color in a species of newt has displayed autosomal linkage, right? So, when we're talking about autosomal linkage, we'll be writing it in brackets like this, right? The gene for tail length has 2 alleles, right? Now, dominant allele T produces a normal length tail, and recessive allele T produces a shorter length tail, right? Gene for scale color has 2 alleles. Dominant allele G produces green scales and recessive allele G produces white scales, right? So we have our tail length genes, alleles, right? Normal length one and shorter length one, and we also have for scale color, right? So basically what we can do here is that we are crossing our normal tail and green scales into short tail and white scales, right? So when I'm talking about normal tail, right, what are gonna be my particular alleles, right? So I'm gonna have TG, capital T and capital G, right? Because it has okay. So because first of all you need to remember one thing, right? Tail length and scale color are what? They're displaying autosomal linkage, right? So they're gonna exist in the parental combination, right? So if you're talking about normal tail and green scales, right? So you'll write one capital T, right? Because normal tail length, and one capital G because that gives you green scales, right? And now the other part is, now that is on one chromosome, right? Now the other part can be another combination can be TG, right? Why? Because T is gonna give you shorter length tail and G is gonna give you white scales, right? So but because that's recessive, that's not gonna express, right? And now another thing is that for our short tail and white scales, again when we look at the particular letters given, we can reduce that TG, right? Basically our short tail is coming from small t, right? And our small g is for white scales, right? So we're crossing them, right? Okay, and another thing is, yes, so a new heterozygous for a normal tail and green scales is crossed with a new that has a shorter tail and a white scale, right? So first of all, because we were talking about heterozygous, one we took dominant alleles and one we took recessive alleles, and why have we put them together in brackets? Because they're autosomal, because they're displaying autosomal linkage, right? And this is crossed with a new that has a shorter tail and white scales, and they have not mentioned heterozygous, so that is why we took both of them recessive alleles, right? Also because G we know is gonna be a recessive allele, but the only problem is that because they're autosomal, they're displaying autosomal linkage, so the tail length allele and the scales allele will be present on the same chromosome, right? So you need to put them in a bracket, right? Now, when you're gonna cross the gametes, you'll see that you'll be getting 1-to-1 ratio, right? So one for normal tail green scales and another for short tail white scales, right? So you can say because there is autosomal linkage, the chromosomes are gonna stay in the original parental combination, and that is why we're gonna have the same phenotype resulting, right? The codominance. When working with codominant alleles, the genetic diagrams can be constructed in a similar way. However, the genotypes are represented using a capital letter for the gene and superscript letters for the alleles, right? So, for example, if I'm talking about blood group, right? Okay, not blood group, but for example, if I'm talking about two alleles that are codominant, right? Yes, we can talk about blood group actually. For example, AB, right? So in that, our A allele for blood group and B allele for blood group, they both display codominant, right? They're both dominant, right? So what we can write it as is that I superscript A and I superscript B, right? So that is how we're going to be displaying codominance. Now moving on to epistasis. Okay, so in some cases, one gene can affect the expression of another gene. Epistasis, when two genes on different chromosomes affect the same feature, right? So two genes on different chromosomes, they're affecting the same feature, right? Now, if epistasis is present, it needs to be taken into account when determining the phenotypes of individuals. The whole combination of alleles from the different genes dictates the phenotype. Now again, we also have a worked example explaining epistasis, right? There is a gene that dictates the feather color of pigeons, right? The gene has two alleles, RR, right? Allele R is going to code for a pigment that produces gray feathers, right? Allele R doesn't produce a pigment, resulting in white feathers. Now another gene has also been found to have an effect on feather color, right? So this gene is going to have two alleles, right? The dominant allele F stops gray feathers being produced even if the allele R is present, right? So basically what's happening here is that you can see that we have two genes on different chromosomes affecting the same feature, right? We also have F alleles and we also have R ones, right? So for example, if we have these are the possible phenotypes, right? Now, when we're talking about these phenotypes, you can see that the dominant allele F was stopping gray feathers being produced, right? So wherever we're gonna have the dominant allele F will not have what? Will not have gray feathers, right? Even if we have allele R present, right? That is how epistasis is gonna affect the phenotype, right? Now predicting inheritance test crosses. A test cross can be used to deduce the genotype of an unknown individual that is expressing a dominant phenotype. And that we just did, that we're going to cross it with the recessive phenotype. If any individual is expressing the recessive phenotype, then the individual in question will have a heterozygous phenotype, heterozygous genotype, right? Okay, now results. For a monohybrid test cross, okay, so it's written for both the crosses that we need to study, okay. So basically in our syllabus we're going to be doing two test crosses, monohybrid and dihybrid. When we're talking about one characteristic, we'll be doing a monohybrid one, right? For example, this normal tail and scales one, because we were talking about two features, it was a dihybrid cross, right? So for a monohybrid test cross, if no offspring exhibit recessive phenotype, then the unknown genotype is homozygous dominant. And if it is gonna express it, then it's gonna be heterozygous, right? And also the results, this we already discussed, but we didn't discuss the result for a dihybrid test cross. So if no offspring exhibit the recessive phenotype for either gene, then the unknown genotype is homozygous dominant for both genes, right? So if we don't have recessive phenotype for either gene, right, then the unknown genotype is homozygous dominant for both genes, right? If at least one of the offspring exhibit the recessive phenotype for one gene, then the unknown genotype is heterozygous for one gene and the homozygous dominant for the other, right? So what's happening is that basically if at least one of the offspring is exhibiting the recessive phenotype for one gene but not the other, right, then the unknown genotype is heterozygous for one gene, right? And if at least one of the offspring exhibits the recessive phenotype for both genes, then the unknown genotype is heterozygous for both genes, right? This is what you need to remember, the results of the test cross, right? Now, predicting inheritance, chi-squared test, right? So, the difference between expected and observed results in experiments can be statistically significant or insignificant, right? Meaning that there may be certain factors, actual factors that account for the difference between the expected and observed results, or they might be just due to chance, right? It is the difference between results is statistically significant. It can suggest something else is happening in the experiment that isn't being accounted for. For example, linkage, right? A statistical test called the chi-squared test determines whether there is a significant difference between the observed and expected results in an experiment. This chi-squared test is completed when the data is categorical, data that can be grouped, right? So basically what we'll be doing is that to do this, we need what? We need our observed and expected values, right? And we're going to calculate the difference, right? And then we're going to square each difference and we're going to divide and we're going to get a sum of that, right? So what we did was we obtained the expected and observed results for an experiment, we calculated the difference between each set of results, right? And then we squared each difference, right? And then we're gonna divide the sum of this square difference, right? So divide each square difference by the expected value and get a sum of these answers to obtain the chi-squared value. That's the last point that we need to do, right? Now, basically, now what's happening is, now you're gonna get a value using this, right? I forgot to mention the question, the equation. That will make it much easier for you. But you can search out the equation for our, the formula rather, for chi-squared test, right? So it will be really easy for you to calculate, right? Now, analyzing chi-squared values, right? Now, whatever your values is, you can compare it with the table that relates chi-squared values to probabilities, right? Now, if the chi-squared value represents a larger probability than the critical probability, then it can be stated that the differences between the expected and observed results are due to chance, right? Now, to determine the critical probability, what you do is that you basically use a probability of 0.05, right? So basically, okay, now first thing that you need to remember is that you need to know about the different phenotypes that you're examining, right? Now, for example, if I am talking about 4 phenotypes, right, I'm talking about, let's say, here's eyes, hair color, eye color, and height, and nose shape, for example, right? Now, I have 4 classes of phenotypes right here, right? So, my degrees of freedom is gonna be 4 minus 1. That's 3, right? Now, that 3, and that is my degrees of freedom, and I'm gonna look for the probability, for my probability, critical probability, and we're going to use a probability of 0.05, right? And now, if, for example, now whatever is the answer, we'll see that if the probability that we're getting for it is smaller, then the idea is that basically then the difference might be due to chance. But if it's greater, then there might be other factors, right? So, okay, if it represents a smaller probability than the critical probability, then the results in, okay, I think I said the opposite, but okay. So if it represents a smaller probability than the critical probability, then it can be stated that the differences between the expected and observed results are due to chance, right? And represents larger, yes. So if there is a larger probability than the critical probability, then the differences are due to chance, and if a smaller probability, then the results are significant, right? I'll just try to search up the chi-square table so that I can explain to you in a better way what I'm trying to say. Alright, so I have searched the table. I'll explain you. Okay, so what you can see in this table here is that, as you can see, we have degrees of freedom and we have the significance level, which is our critical probability, which has a critical probability that we're considering 0.05, right? So for example, I have a degrees of freedom 3, and now I'll look for the corresponding value at 0.05. So that is coming as 7.815. Now for example, now I'll compare this value with the value that I calculated from my chi-square test, right? So for example, my value is coming as 0.252, right? Now, I'll see that the probability for 7.815 is 0.05. So for a value of 0.252, my probability will go towards the left hand side, right? That means my probability is becoming bigger, right? It's gonna be bigger than 0.05, right? So when my probability is bigger than 0.05, I can deduce that my differences between observed and expected results are just due to chance, right? Alright, so continuing with our chapter, genes, proteins, and phenotype. A gene can affect the phenotype of an organism, and we know that a gene is going to code for a single protein, right? The protein affects the phenotype through a particular mechanism, right? The phenotype of an individual can also be affected by the environment. Genes code for mRNA, which gets translated into a polypeptide protein that affects the phenotype of the organism, right? So basically, we'll be talking about a particular mechanism, right, by which a protein can affect the phenotype, right? So first thing is TYR gene and albinism. So humans with albinism are going to lack the pigment melanin in their skin, hair, and eyes, right? And this causes them to have very pale skin, pale hair, and pink irises, right? Now, there is a metabolic pathway to produce melanin, right? So basically, the amino acid tyrosine is converted to DOPA by the enzyme tyrosinase, and then the DOPA will be converted to dopaquinone again by the enzyme tyrosinase, right? And then that dopaquinone is converted to melanin, right? So we have the sequence as follows: tyrosine being converted to DOPA, then the DOPA being converted to dopaquinone, and then that particular thing being converted to melanin, right? So a gene called TYR located on chromosome 11 codes for the enzyme tyrosinase, which is basically metabolizing this pathway, right? There is a recessive allele for the gene TYR that causes the lack of the enzyme tyrosinase or the presence of inactive tyrosinase. Without the tyrosinase enzyme, tyrosine cannot be converted into melanin, right? Then we have another example. We have HBB gene and sickle cell anemia, right? Sickle cell anemia, we know, is a condition that causes individuals to have anemia, which is, breathlessness, lack of oxygen, right? Episodes of pain, frequent infections, right? Now, humans with sickle cell anemia have abnormal hemoglobin in their red blood cells, right? Now, beta globin is a polypeptide which is found in hemoglobin that is coded for by the gene HBB, which is found on chromosome 11, right? So basically, we're talking about a polypeptide that is found in hemoglobin, which is coding for the gene HBB that is found on chromosome 11, right? And there is an abnormal allele for the gene HBB, which is going to produce a slightly different amino acid sequence to the normal allele, right? So the change of a single base in the DNA of the abnormal allele results in an amino acid substitution, right? So basically what happens is that the base sequence CTC is replaced by CAC, right? Now that has changed the nucleotide sequence, right? Which means that now it's coding for an abnormal allele, right? So this change in amino acid sequence replaces the amino acid glue with VAL, right? So this is going to result in an abnormal beta globin polypeptide, right? So the abnormal beta globin in hemoglobin affects the structure and shape of the red blood cells. So now they're going to have a half-moon shape, right? So they'll be unable to transport oxygen around the body. They will stick to each other, clump together, blocking capillaries, right? So a homozygous individual that has two abnormal alleles for the HBB gene produces only sickle cell hemoglobin, right? And if there is going to be a heterozygous individual, then they're going to be one normal allele and one abnormal allele. So we're going to have both the normal hemoglobin and sickle cell hemoglobin, right? So these people will be carrier of the disease, however, they may have those symptoms, right? Then moving on to the next example, we have F8 gene, factor 8 gene, and hemophilia, right? The factor 8, we just studied that it's basically a coagulating agent that plays an essential role in blood clotting, right? So the factor 8 gene codes for the factor 8 protein, right? Now, there are abnormal alleles of the factor 8 gene that result in production of abnormal forms of factor 8, less production of normal factor 8, and no production of factor 8, right? A lack of normal factor 8 prevents normal blood clotting and causes the condition hemophilia, right? And we know that the factor 8 gene is present on the X chromosome and it's a sex-linked gene, right? So hemophilia is also a sex-linked condition, right? And we also studied of how if males have an abnormal allele, they will have the condition as they have only one copy of the gene. However, because females are heterozygous, they may be carrier, they may be suffering from hemophilia, right? Or they may not suffer also, right? So these are the three phenotypes that will result for females, right? Now moving on to HTT gene and Huntington's disease, right? Now Huntington's disease is a genetic condition that develops as a person ages, right? Now usually this disease will only start showing symptoms after a person is 30 years old, right? So an individual with the condition experiences neurological degeneration. That is that they're going to lose their ability to walk, talk, and think, right? And this disease is ultimately going to prove fatal, right? Now, it has been found that individuals with Huntington's disease have abnormal alleles of the HTT gene. Now, the HTT gene codes for the protein Huntington, right, which is involved in neuronal development, right? Now, people that have a large number of repeated CAG triplets present in the nucleotide sequence of their HTT gene suffer from the disease, right? The abnormal allele is dominant over the normal allele, right? So, even if there is one gene present, right, the person is gonna suffer from the disease, right? Now, moving on to the next pointer, the role of gibberellin stem elongation, right? In some plant species, their height is partially controlled by their genes. The LE gene dictates the height of some plants, right? It has two alleles, right? So we have the dominant allele, which is going to give you tall plants, and we also have the recessive allele, which is going to give you shorter plants, right? Now, the gene is going to regulate the production of an enzyme that is involved in the pathway that forms active gibberellin, right? So the gene is basically helping you for what? For active gibberellin, right? And active gibberellin is basically a hormone that is going to help the plants grow by stimulating cell division and elongation in the stem, right? And the recessive allele is going to result in a non-functional enzyme, right? So basically what's happening is that we have a gene, right? And that gene is coding for an enzyme that is involved in the pathway to produce active gibberellin, and gibberellin is the hormone that stimulates cell division and elongation in the stem, right? So the recessive allele is one nucleotide different to the dominant allele, right? Basically, there is a single amino acid substitution taking place through need being replaced by alanine, right? So this changes the primary structure of the enzyme, right? That means that active gibberellin cannot be produced, right? So the plants will not be able to grow tall, right? So plants that are homozygous for the recessive allele, LE, are dwarfs, right? And the ones so basically farmers would have to apply active gibberellin to shorter plants to stimulate growth, right? Now moving on to gene control. The nucleus of every cell in the human body contains the same genes. However, not every gene is expressed in every cell, right? Not all of these genes are expressed all the time, right? There are several mechanisms that exist within cells to make sure that the correct genes are expressed in the correct cell at the right time, right? So this is what's important, and gene control is important, right? That the correct gene is being expressed at the right time, right? So there are two types of genes. We have regulatory genes and we have structural genes, right? A structural gene is going to have a function within a cell, codes for a protein that has a function within a cell, right? Now, for example, we have the factor VIII, which is involved in blood clotting. And a regulatory gene is going to control, it codes for a protein that controls the expression of another gene, right? Now, another thing that you need to know about is that we have inducible and repressible enzymes, right? So, inducible enzymes would only work if their substrate is present, right? Now, the presence of the substrate induces the synthesis of the enzyme by causing the transcription of the gene for the enzyme to start. Repressible enzymes are synthesized as normal until a repressor protein binds to an operator, right? Now, this will be doing in detail, that repressible enzymes will be the ones which will be synthesized as normal until a repressor protein binds to an operator, right? So when the repressor protein is going to bind to an operator, transcription cannot take place, right? So this is what we'll be looking at, right? And we're doing this, gene control is important so that energy is not wasted when it's not required, right? For example, if the substrate is not present, then the enzyme wouldn't be required, right? Now, a very good example of this is the lac operon, right? Now, operon is basically a cluster of genes that will be working together, right? Okay, so regulatory genes control structural genes and their levels of protein production. Regulatory genes sometimes have control over several structural genes at once, right? So we're going to form an operon, which will be a group or a cluster of genes that are controlled by the same promoter, right? So the lac operon found in some bacteria is one of the most well-known of these. The lac operon controls the production of the enzyme lactase, right? Also called beta-galactosidase. Alright, so lactase, also called beta-galactosidase, right, and two other structural proteins, and their production is going to be controlled by the lacoperon, right. Now the lactase is going to break down the certain lactose so that it can be used as an energy source in the bacterial cell, right, and it will be an inducible enzyme, meaning it will only be synthesized when lactose will be present, right, when its substrate, which is lactose, will be present, right. Now, moving on to the structure of the lacoperon, right? Now, first of all, we will have a promoter for structural genes, then we'll have the operator. Okay, now the structural gene LAC-C is going to code for lactase, LAC-Y is going to code for permease, which is going to allow the lactose to enter into the cell, right? And then the structural gene LAC-A is going to code for trans-acetylase, right? Now, located to the left of that particular promoter will have the promoter for the regulatory gene, right? And the regulatory gene LAC-CL that codes for the LAC repressor protein, right? Now the LAC repressor protein, now this is a really important point to remember, right? That the LAC repressor protein has two binding sites and allowed to bind to the operator in the lacoperon and also to lactose, right? So it can either bind to the operator or either lactose can bind to the repressor protein, right? And this thing is important in controlling the production, right? So when it's going to bind to the operator, it's going to prevent the transcription of the structural genes, right? Because RNA polymerase will not be able to attach to the promoter, right? But when it's going to bind to the lactose, it will distort the shape of the repressor protein so that it cannot bind to the operator, right? Now when it cannot bind to the operator, transcription can take place as usual, right? Now, when lactose is absent, the following processes take place, right? The regulatory gene is transcribed and translated to produce LAC repressor protein, right? Now, this protein is going to bind to the operator region, right? And because it's bind to the operator region, RNA polymerase cannot do transcription, right? So, no lactase enzyme. And when lactose is going to be present, what's going to happen is that our lactose is going to bind to the second binding site of the repressor protein, right? So its shape is distorted, meaning it cannot bind to the repressor site, so RNA polymerase is going to continue transcription, right? So mRNA from all three structural genes is translated, right? So enzyme lactase is produced and lactose can be broken down and used for energy by the bacteria, right? Lactose binding to the repressor protein, which frees up the operator region of the lacoperon, so RNA polymerase can bind and begin transcription of the structural genes. So to give you an idea of how the operon looks like, I've searched the picture, right? So you can see here that we have LAC-CL, which is our regulatory gene. We can see the promoter for the regulatory gene. We can see the operator, promoter for structural genes, and then the three structural genes as well, right? So this is very important because this comes in the paper for labeling, right? So need to know, right? Next, what we have is gene control transcription factors, right? So prokaryotes, we just talked about lacoperon in bacteria, right? So prokaryotes use operons to control the expression of genes, right? But eukaryotes are going to use transcription factors, right? So a transcription factor will be a protein that will control the transcription of gene by binding to a specific region of DNA, right? Now, they show that genes are being expressed in the correct cells. Again, gene control, right? It is estimated that 10% of the genes code for transcription factors. There are several types of transcription factors that have varying effects on gene expression. There is still a relatively young area of research and scientists are working hard to understand how all the different transcription factors function, right? But basically, transcription factors are going to allow organisms to respond to the environment. They'll also help some hormones achieve their effect, right? So how they will be working is that they're going to bind to the promoted region, right? Now this binding can either allow or prevent the transcription of the gene from taking place, right? So the presence will either increase or decrease the rate of transcription, right? Now we'll be looking at an example of PIF, right? PIF is a transcription factor that is going to activate the transcription of the amylase gene, right? So this transcription factor, which is PIF, is going to bind to the promoted region of a gene, which will allow RNA polymerase to bind, right? So that is going to allow transcription to occur in this case, right? Now, the example that we'll be studying is gibberellin, right? So, plants use transcription factors in a similar way to animal cells, right? Gibberellin, we know, is a hormone that is going to control seed germination by stimulating the synthesis of the enzyme amylase, right? Now, it does this by influencing transcription of the amylase gene, right? So, when gibberellin is going to be applied to a germinating seed, there is an increased amount of the mRNA for amylase present, right? So basically, what is the mechanism behind this? What happens is that there is a protein called DELA, right? Now, this protein is going to be broken down by gibberellin, right? And this is necessary for the synthesis of amylase, right? Now, what do we require? What are the components? We have the repressor protein DELA, we have the transcription factor PIF, we have the promoter of the amylase gene, and gibberellin, right? Now, first of all, the repressor protein, which is DELA, is going to bind to PIF, which is the transcription factor, right? Now, that is going to prevent it from binding to the promoter of the amylase gene, so no transcription can occur, right? However, if we have gibberellin, right? Gibberellin was doing what? It was causing a decreased amount of the mRNA for amylase to be present, right? And it was stimulating the synthesis of the enzyme amylase, right? So basically, how is it doing that? It binds to the gibberellin receptor, right? And enzyme which starts the breakdown of DELA, right? So basically, when gibberellin is going to bind to the gibberellin receptor, right, that's going to start the breakdown of DELA, right? Now, when PIF, which is the transcription factor, is no longer bound to the DELA protein, right, now the PIF, which is the transcription factor, that can bind to the promoter of the amylase gene, right? So now the transcription of amylase can begin and our amylase will be produced, right? Now the breakdown of DELA protein by gibberellin allows the transcription factor PIF to bind to the promoter for the amylase gene and for transcription to initiate, right? Now this was all for this lecture, and I'll see you in the next one, and I hope you found this video useful.